Now, to start our presentations, it's my pleasure to welcome for opening remarks Dr. Leo Bouthillier, Director, Center for Blood Products, and Biotherapeutics, Biologic and Genetic Therapies Directorate, Health Products and Food Branch, Health Canada. Please join me to welcome Dr. Bouthillier.
Thank you very much for the introduction and good day everyone. I was very pleased to see that we had over 3,000 registrants for this webinar, and they are from all over the globe as I understand, which is great. I would like to begin by saying that ICH gave itself a mission of improving health, and this has been true since it began in 1990. Today, the mission seems more important than ever as we face climate change, coming out of a global pandemic, as well as regional military conflicts, all of which with impacts on the global supply chain and on drug development. There is just a growing need for efficient, continued development and delivery of drugs to patients who need them for a plethora of conditions or diseases, some of which can cause reduced quality of life, chronic disabilities, and even premature death. ICH debut was in 1990. But it is only after the reform of 2015 that it really expand the organization to include a really diverse and wider membership. It began with 6 members representing regulators and industry and 4 observers. Today, we have 21 members and 37 observer organizations. We have also approximately 30 expert working groups at any given time that involves over 1,000 people. So that is a lot of people, and it's quite amazing actually. ICH has therefore become an incredibly large and productive endeavor to produce modern tools and approaches to drug development. And that includes clinical studies and modernizing manufacturing approaches. The range of topic is very impressive. Thank you very much for the introduction and good day everyone. I was very
effectiveness and quality. It accomplished this goal by convening experts.
A very rich program starting with an overview of ICH, followed by presentations on a wide range of topics in development. Namely, there will be presentations on drug interaction studies, post-approval safety data management, good clinical practice, general principles on plan, design, and analysis of pharmacoepidemiological studies that utilize real-world data for safety assessment of medicines, analytical validation and analytical procedure development, viral safety evaluation of biotechnology products derived from cell lines of human or animal origin, quality risk management, and finally, a presentation on exploratory work on cell and gene therapies. All of these areas are really essential to continue to develop safe, effective, and high-quality medicines to patients that need them. And so we are very pleased that you could join us today, and I want to thank you again for taking the time out of your busy days and look forward to the presentation and discussions. With that, I would like to turn it over to Jill Adelberg from the US FDA, who will provide a more detailed overview of ICH. Over to you, Jill.
Leo, thanks very much for that warm welcome. For those who are unfamiliar with ICH, I'm going to provide a brief overview of the organization, its approach, and its process for harmonizing international drug regulation standards. ICH was established more than 3 decades ago by representatives of the regulatory agencies and industry associations from the US, Europe, and Japan. From its start, Health Canada, Swissmedic, and the World Health Organization also provided valuable contributions. ICH seeks to develop uniform, scientifically driven standards for medicines that enhance their safety, effectiveness, and quality. It accomplishes this goal by convening experts to work on development or revision of harmonized technical guidelines and standards implemented by regulatory authorities around the world. These standards guide the clinical development, safety assessment, control and manufacture of high-quality medicines, and replace a patchwork of duplicative individual country requirements. In turn, these standards yield efficiencies that improve patient health. In 2015, ICH extended the reach of harmonization beyond its founding members, becoming a nonprofit legal entity under Swiss law. The ICH Association opened the door to the wider community of pharmaceutical regulators and global industry stakeholders, and has enriched ICH's work by offering a broader range of perspectives. ICH has grown to include 21 regulatory and industry association members spanning 6 continents and 37 observers representing more than 2/3 of the world's population. In the past year, ICH welcomed Egypt as its newest member, and Nigeria and Hong Kong as new observers. ICH is governed by the Assembly, the overarching body, including all ICH members and observers. The ICH Assembly approves new topics for harmonization, adoption, amendment, or withdrawal of ICH guidelines, admission of new members and observers, election of Assembly and Management Committee leaders. The Management Committee is a smaller body, including permanent and standing members and elected members, that oversees operations, including administrative and financial matters and working group efforts. It recommends to the Assembly selection of new topics for harmonization and adoption, withdrawal, or revision of guidelines. This slide maps the ICH governance structure. At the center is the Assembly. The ICH and Measure Management Committees branch to each side. The Secretariat provides day-to-day management of ICH, including preparing and documenting ICH Assembly and Management Committee meetings. ICH coordinators are selected by ICH members and facilitate the efficient operation of ICH harmonization activities. They serve as the central point of contact for their organizations with the ICH Secretariat and facilitate conversation between the ICH Management Committee and the working groups. Over its history, ICH working groups have produced and updated over 70 harmonized technical guidelines on all aspects of human drug development. More are currently under development. Existing guidelines are also regularly updated and revised as science and digital platforms evolve. ICH pioneered development of a harmonized marketing application accepted by regulators in all ICH regions. The Electronic Common Technical Document has dramatically reduced the burden for sponsors seeking marketing approval, who can now submit data and information organized in the same way to each regulator. ICH also developed the Medical Dictionary for Regulatory Activities Terminology, or MEDRA, to facilitate sharing of regulatory information internationally. MEDRA is rich and highly specific standardized medical terminology used for registration, documentation, and safety monitoring of medical products. Since its launch in 1999, MEDRA has steadily grown. It's now available in more than a dozen languages and counts over 6,600 organizations in more than 125 countries among its subscribers. ICH also produces question and answer documents and training materials to support implementation of the guidelines, as well as reflection papers that provide a more strategic approach to development of new topic proposals. This slide provides a sample of harmonized topics considered by ICH working groups. Among the 70 guidelines referenced earlier are foundational standards on such topics as good clinical practices for the conduct of clinical trials to safeguard protection of trial participants and the reliability of results. We're going to hear a little bit more about this working group, E6R3, which is currently developing underlying principles to remain relevant as technology, communications, and science rapidly evolve across healthcare. Among the 70 guidelines is also good manufacturing practices that specify technical requirements for medicines, including analytical validation, and that describe tools and approaches to support continual improvement after approval, and also included our standards for the exchange of meaningful information about potential side effects of medications to facilitate signal detection and safety monitoring and to reduce patient harm. ICH considers proposals for new harmonized guidelines on an annual basis. The cycle begins in the fall and ends with the approval of new topics by the Assembly in the spring. In between, the Management Committee evaluates the proposals and provides a recommendation on new topics to the Assembly. Any ICH member or observer may submit proposals for new topics. The topics are prioritized based on factors including the public health value, the degree of scientific consensus, and the availability of expert resources to support development of these topics. When the Management Committee recommends a new topic for Assembly approval, the party who submitted the proposal develops a concept paper outline for Assembly consideration. Approval of the concept paper outline may occur concurrently with the new topic proposal or soon after. Once the concept paper outline is approved by the Assembly, ICH establishes an informal working group of experts to develop a concept paper. A concept paper describes the perceived problem and the issues to be resolved by a harmonization project and outlines the estimated timeline for meeting key milestones. It defines the scope and parameters of the guideline and guides its development. Within ICH, when all informal working group members reach consensus on a concept paper, it is submitted to the Management Committee for approval. Once approved by the Management Committee, an expert working group is formed to develop the harmonized guideline based on the objectives in the concept paper. In practice, informal working group members typically transition to membership on the expert working group. Harmonized guidelines are developed in a 5-step process. Typically, it takes approximately 3 years to complete development of a new guideline or a significant revision to an existing guideline, though these timeframes can and do vary based on the topic. At step 1, an expert working group engages in the substantive work of developing a consensus draft of the guideline. When the draft is completed, the topic leads for each party that are represented on the working group sign off on the guideline for their organizations. Step 2 is a 2-step process. First, the Assembly endorses the draft guideline. Second, regulatory members of the Assembly separately do so. At step 3, the guideline moves to a public consultation phase in which it's submitted for public comment in a transparent process designed to solicit thoughts from a wide array of stakeholders. The expert working group collects all of the comments received, reviews and evaluates them, and revises the draft guideline into a final guideline. And finally, at the final stage of step 3, the working group's topic leaders sign off on the final guideline. At step 4, regulatory members of the Assembly approve the final guideline. And finally, at step 5, ICH regulatory members and observers implement the final ICH guideline within their respective country or region, achieving the goals of harmonization. The status of implementation efforts is published on ICH's website, and it's updated periodically by the ICH Secretariat. As you can see from this chart, over 700 technical experts have participated in guideline development in recent years. More than a third of these experts come from recent ICH members and observers, reflecting a significant level of interest and engagement by ICH member parties. As part of its effort to enhance international acceptance of harmonized standards, ICH develops high-quality training materials to educate audiences about the scientific and regulatory principles that are outlined in the guidelines and to help translate them into predictable regulatory practice. Among ICH's training efforts include a training library which is posted on the ICH website, funding support for training programs organized by ICH regulatory members and observers, an ICH recognized training program, which is hosted by a variety of organizations, associations, regulatory authorities, and by academia. These programs are offered in person, virtually, and online, and information is provided on the ICH website. And other online training materials, including some translations, are also posted on the ICH website. A number of topics previously approved by the ICH Assembly are scheduled to get underway soon. They include a revision to the Q6A and Q6B guideline on specifications. This guideline revision was recommended by ICH's Quality Discussion Group. Also included is a general considerations for patient preferences studies, which is recommended in an ICH reflection paper on patient-focused drug development. Also starting soon is a non-clinical safety studies for oligonucleotide-based therapeutics guideline, and a structured product quality submissions guideline. And work on the structured product quality submissions guideline is slated to begin when the M4Q R2 guideline reaches step 2 later this year. For a quick recap, ICH is a unique organization that leverages the expertise of scientific experts in both government and in industry to develop common international guidelines and standards. It employs a transparent, science, and consensus-based process, and it includes a commitment by ICH regulatory members to implement ICH guidelines, and this has been a core operating principle since the organization's inception. I hope this introduction has been helpful. Now, thank you very much for your time, your attention, and your interest in our efforts. I'm going to turn our program over now to our first panel of presenters who will be highlighting efficacy guidelines that have reached or are expected soon to reach important milestones. Our presenters include Dr. Kelly Reynolds, FDA's topic lead on the M12 Drug Interaction Studies Guideline, which is nearing completion. Kelly is the director of the Division of Infectious Disease Pharmacology in CDER's Office of Clinical Pharmacology. Next up will be Dr. Craig Zinderman. Craig is FDA's topic lead on E2D R1, post-approval safety data management, definitions and standards for management and reporting of individual case safety reports. The draft guideline was just posted on ICH's website, having been approved at Step 2A and B earlier this month. Craig serves as the Associate Director for Medical Policy for CBER's Office of Biostatistics and Pharmacovigilance. And last but not least will be Dr. Carol Legaret, who serves as Health Canada's topic lead during the development of the draft E6R3 Good Clinical Practice Principles in Annex 1, which was published last May. Carol is a senior advisor in the Office of Clinical Trials for Health Canada's Pharmaceutical Directorate, Health Products and Food Branch. So without further ado, Kelly, the floor is yours.
Hello. Today I'll provide an update on the ICH M12 guideline, which addresses drug interaction studies. As you will hear during the presentation, the expert working group is finalizing edits based on comments received during the public consultation period. I will start with a background that includes general principles related to evaluation of drug interactions, the objectives and scope of the guideline, and the timeline for the expert working group from start to finish. The table of contents displays the full scope of topics included in the guideline. Due to the number of topics included in the guideline, I will focus on 5 topics that generated discussion at various points during the working group's deliberations. The topics are in vitro cutoff values, drugs with high protein binding, endogenous biomarkers, studies with concomitant medications, and the interpretation of study results. General principles that support the development of the guideline are as follows. Drug interactions can occur when patients take more than one drug, which may impact safety or efficacy, resulting in altered risk-benefit. The evaluation of drug interaction potential should be risk-based and proceed in a stepwise manner from in vitro to clinical, and it often includes predictive modeling. The evaluation should begin early in drug development to avoid unnecessary exclusions in clinical trials and to ensure the safety of clinical trial participants. The timing and utility of non-clinical studies, clinical studies, and predictive modeling is dependent on clinical context and the type of product. A common consideration is the intended patient population and other drugs they need to take. The interpretation and translation of drug interaction study results should be based on an understanding of the variability of the drug exposures and the exposure-response relationships for desirable and undesirable drug effects. The objectives of this guideline are to develop recommendations that promote a consistent approach in designing, conducting, and interpreting in vitro and clinical drug interaction studies during development of a therapeutic product, and to reduce uncertainty for the pharmaceutical industry to meet the expectations of multiple regulatory agencies, which may lead to more efficient use of resources. There's a wide range of potential topics related to the scope of drug interaction evaluations during drug development. The scope of this guideline includes pharmacokinetic interactions with a focus on enzyme and transporter-mediated interactions, and it focuses on small molecules and biologic products, and the specific biologic products are monoclonal antibodies and antibody-drug conjugates. The guideline does not address pharmacodynamic interactions, pharmacokinetic interactions due to gastric pH change, formation of complexes or chelates, or food effect. And it does not address interactions with new modalities such as oligonucleotides. The working group selected the scope based on topics in the greatest need of global harmonization and for which adequate scientific information is available to provide detailed instructions. The drug interaction topic was proposed in November 2018. The initial work started in June 2019 with the formation of the expert working group at the 2019 meeting in Singapore. Due to the pandemic and travel restrictions, the working group conducted all meetings virtually in 2020 through 2022. We collaborated effectively in spite of the virtual setting and the time difference. The draft guideline was published in May 2022. With public consultation period ending that November. 2023 was an active year with comments discussed when we met in Geneva and finalization of the guideline revision soon after our meeting in Prague. We are starting the various clearance steps and we anticipate adoption of the guideline in March. In addition to the guideline, we are working on a question and answer document and educational materials to support the implementation. During the public comment period that closed in November of 2022, 30 stakeholders provided approximately 1,000 comments, and the comments were distributed across the various sections. The introduction to the guideline includes the objective, background, scope, and general principles that support the details outlined in the guideline. The in vitro evaluation section describes the process for evaluating metabolism-mediated interactions, transporter-mediated interactions, DDI potential of metabolites in vitro. This section includes cutoff criteria for determining whether the in vitro study results translate to a need for clinical evaluation of drug interactions. The clinical evaluation section describes various types of clinical studies and considerations when planning the studies. I want to point out that we include information about an emerging topic, endogenous biomarkers, and I will describe that topic later in my presentation. The guideline provides information about pharmacogenetic variations in drug metabolizing enzymes and drug transporters to the extent that they affect evaluation of drug interactions. And there is a brief section that describes drug interaction considerations for therapeutic proteins. There is a section that describes reporting and interpretation of clinical drug interaction study results, which directly informs risk assessment and management. The detailed appendices support the studies and analyses described in the main body of the guideline. The glossary is a new appendix that defines terms used in the guideline. Another new appendix provides details about the evaluation of protein binding because the accuracy of the methods is essential for interpretation of in vitro results. Other appendices describe in vitro methodology for metabolism and transporter studies, predictive modeling, and it provides a list of drugs that can be used in vitro and clinical studies. Finally, we provide a list of references that support emerging areas and key topics. However, it is not an exhaustive list of references. With that overview of the contents of the guideline, I will use the rest of the presentation to focus on 5 topics: in vitro cutoff values, considerations for drugs that are highly bound to plasma proteins, endogenous biomarkers, clinical studies with likely concomitant medications, and the interpretation of study results. Cutoff values compare an in vitro measure of inhibition or induction with an estimated clinical exposure to determine whether a clinical drug interaction study is recommended. The cutoff values are of high interest to drug developers because they directly affect the number of clinical drug interaction studies needed. The working group considered the following factors when selecting cutoff values: consistency among regional guidelines, in vitro in vivo analyses based on literature and FDA and EMA approved products, the impact of capping protein binding estimates for drugs that are highly protein bound, and the likelihood of false negative predictions. The next topic is the handling of drugs with high protein binding. The draft guideline indicated the following regarding protein binding measurements. There are uncertainties in the accuracy of protein binding measurements for highly bound drugs, those with greater than 99% protein binding. Due to uncertainties, the fraction unbound in plasma should be capped at 1%. However, it also indicates that there have been advances in the methodology and it's an active area of research. Thus, in some situations, measured fractions less than 1% can be used if the accuracy and the precision of the measurement is demonstrated, and this includes the provision of validation data, including bioanalytical method and appropriate positive controls. Based on comments received during the public consultation, the working group discussed the need for additional clarity regarding expectations related to bioanalytical methods, when positive controls are needed, and whether multiple protein binding experimental approaches are needed. As we revised the guideline, we invited external experts from industry to present protein binding evaluation results. We also conducted in-depth literature review on the topic. Following this work, we revised the guideline for clarity and added an appendix that describes considerations for protein binding measurements for highly bound drugs. Endogenous biomarkers is another emerging area that the guideline addresses. The draft guideline indicated the following regarding the use of endogenous biomarkers. Recent literature describes the potential utility of endogenous substrates for some drug transporters. Evaluation of change in exposure of the endogenous substrate in the presence of investigational drug may provide information about the drug's potential as a transporter inhibitor. Based on comments received and continued advancements in this emerging area, the working group discussed the need for additional information in the guideline. Similar to the work for protein binding, we invited external experts from industry to present results from their evaluation of this topic, and we reviewed recent literature. In the end, we revised the guideline to clarify how this emerging area can contribute to drug interaction evaluation. During the consultation period, we received a range of comments requesting more clarity on clinical studies with likely concomitant medications. As a reminder, the draft guideline indicated that drugs with a well-understood and predictable pharmacokinetic and drug interaction properties regarding level of inhibition, induction, or metabolic pathway are known as index drugs. Studies with likely concomitant medications often follow index studies. The specific concomitant drugs for evaluation are selected based on mechanistic understanding of the potential for drug interactions and the relative frequency of co-administration. The studies are often informative to patients and medical professionals, but the results may be difficult to extrapolate to other drugs. Currently, we have identified index drugs for CYP enzymes, but not transporters or UGT enzymes. Thus, drug interaction studies for these pathways often include concomitant medications. Based on comments requesting more clarity on specific studies with concomitant medications, the working group further discussed the topic. In general, the selection of potential concomitant medications for drug interaction studies is case by case, and it depends on the therapeutic area, the intended population, and the safety and efficacy properties of the drugs. We consulted with an external expert to refine the list of UGT substrates and inhibitors included in the guideline appendix, and we conducted literature and database research to refine the list of transporter and UGT substrates and inhibitors included in the guideline appendix. The last topic I will discuss today is the interpretation of study results. While writing the draft guideline and addressing comments, this was a topic of high interest. The guiding principle is an emphasis on the use of exposure-response information to determine no-effect boundaries for the drug as an object. An object is a drug that is affected by an inhibitor or inducer. The term victim has been used previously. No-effect boundaries represent the interval within which a change in systemic exposure measure is considered not significant enough to warrant clinical action. The point estimate of the ratio, so with or without the inhibitor or inducer, is normally evaluated in relation to the no-effect boundary, but variability should also be taken into consideration. In the end, we consider all available evidence when interpreting the results. I will close my presentation by acknowledging the numerous individuals who worked on the guideline over the past 5 years. We also appreciate the time that external groups and individuals spent providing valuable comments on the draft guideline and the work of subject matter experts at various regulatory agencies and companies. We are looking forward to the implementation of the guideline. Thank you for your attention, and I look forward to questions during the question and answer period.
Thanks for the opportunity to present an update on the ICH E2D guideline, E2D R1. I'm Craig Zinderman, Associate Director for Medical Policy in the Office of Biostatistics and Pharmacovigilance in FDA's Center for Biologics Evaluation and Research. I'll start with an overview of what the E2D guideline is about, although in brief, it discusses the management and reporting of post-approval safety information. And then we'll talk about some background information on why it was decided to revise the guideline. And then we'll highlight some of the biggest changes that we've made in this revised version of the guideline, including some key new definitions and terminology, the different types of individual case safety reports in the post-approval setting, spontaneous reports and solicited reports. And then most importantly, we'll go through the different sources of safety information, things like digital platforms, internet, patient support programs, literature, and we'll talk about what's been changed in this revised version of the guideline for each source. And then last, we'll cover some scenarios that marketing authorization holders observe in post-approval safety data other than adverse events or adverse drug reactions. And we'll talk about how we've expanded that section on other observations. So what is E2D? The title of E2D is Post-Approval Safety Data, Definitions and Standards for Management and Reporting of Individual Case Safety Reports. This document provides some standardized procedures for post-approval safety data management and the guidance for gathering and reporting this information. It basically provides some of the foundational definitions and concepts and guidance for how MAHs collect and report post-approval data. For example, it defines an adverse event, an adverse drug reaction, and what qualifies as serious AEs or ADRs. And there's definitions for several other fundamental terms that we use in post-market adverse event surveillance. It also defines the timeline for submitting expedited ICSRs. I should note that the original E2D guideline is limited to expedited ICSRs, and it talks about expedited in the title. In this new revised guideline, we've expanded the scope to include all ICSRs, and we've changed the title accordingly. The fundamental concepts and guidance provided in the guideline really apply more broadly to all ICSRs and not just expedited cases. ETD also goes on to discuss various sources of post-approval safety data, for example, information from the internet or information from literature or spontaneous reports that are communicated directly to an MAH and various other sources, and how cases from each of those sources should be managed. So by way of background, the original ICH ETD guideline was adopted as an approved guideline in 2003. So it's more than 20 years old. There's been a bunch of new post-market safety information data sources that have emerged, or at least are used much more often, more frequently today than they were back in the early 2000s. And these new data sources vary in their characteristics and their contribution to post-market safety surveillance. They could have a different level of quality in how much they contribute to what we know about the safety of products, yet they can be really large volume and large burden for marketing authorization holders and regulators alike. Things like patient support programs are used a lot more frequently today than they were in the early 2000s. And then, of course, social media is much more widespread and a source for post-market safety data about medical products like anything else. So it was determined that the definitions and regulatory guidance in the original guideline, which didn't really cover these things like patient support programs and digital platforms or social media, is no longer sufficient to provide guidance on current pharmacovigilance practices. And so an ICH E2D expert working group was formed in 2019 to revise E2D so that it supports more appropriate and more current post-market safety surveillance. The recommendations in the revised guideline are harmonized to the extent possible, but there are a number of differences in post-market safety reporting requirements across the ICH regions. So it wasn't always possible to have guidance that's consistent in every region. Some of these requirements in different areas have changed since the original guideline in 2003. So in places in the guideline where it's needed, where it's applicable, the guideline notes where local and regional requirements may vary, and as such, marketing authorization holders should refer to those local and regional requirements with respect to that particular concept or term. But despite these regional variations, the expert working group believes that the revision, the ETDR1, establishes a harmonized framework for current best practices for managing post-approval safety data, particularly around the reporting terminology and the emerging new sources of data like patient support programs and digital platforms. We're going to talk more about those in a moment. We should note that these slides highlight only the significant updates and changes made in the revised version, the R1 version, and that you should refer to the complete document for all other editorial changes and updates. The expert working group had fairly wide representation across both regulators and industry, with 9 regulators and 5 different industry subgroups participating. The plenary working party for this expert working group includes MHRA from the United Kingdom and Health Canada. There were several brand new definitions added to the revised version of the guideline, and I'm going to highlight some of the key new definitions. One of those is an explicit definition for the term individual case safety report, or ICSR. This is a term that appears in the old guideline, but only once in reference to E2B, which is the ICH standard for electronic transmission of post-market cases to regulators. But the term ICSR wasn't as widely used in that time in 2003, and it wasn't used. It didn't feature prominently in the original guideline, and it didn't have a definition in the original guideline. The new version provides an explicit definition, a description of an adverse event or adverse drug reaction in an individual patient at a specific point in time. This definition also lists 4 minimum criteria for a case to qualify for reporting to regulatory authorities. And those include a suspect product, an adverse event or adverse drug reaction, an identifiable patient, and an identifiable reporter. The revised guideline also includes explicit definitions for expedited reports that are submitted within 15 days, and primary source. The primary source is the individual who provides information about the case to the MAH or the marketing authorization holder. Some other key definitions were added to support the management and reporting of cases from some of the newly emerging sources of post-approval safety data that are increasingly encountered by MAHs. Digital platforms, for instance, is defined in the new guideline as the software and technology used to enable transmission of information between users. And this term, digital platforms, is used in the revision in place of the term internet in the original guideline. It's a broad definition, and it includes, but it isn't limited to, social media, websites, internet forums, chat rooms, and software applications. ODCS is also defined, and this includes clinical trials, registries, and patient support programs, among other things. Note that in the revised guideline, the definition includes a list of basic documentation that MAHs should have for ODCSs that they're conducting. There are also new definitions of patient support program and market research program, and we'll talk about these more in a moment. All 4 of these concepts were areas of specific focus for the EWG in coming together to do the revisions for E2DR1. Section 3, the types of ICSRs, is a new section in the new guideline. The concepts of spontaneous reports and solicited reports used to exist in the section of the guideline on sources of post-approval safety data, but it was moved to its own section because spontaneous and solicited, these concepts really refer to the context in which an ICSR is gathered rather than a specific source of data. Spontaneous reports refers to direct communications by a healthcare provider or consumer to a regulatory authority or an MAH that is not gathered as part of an ODCS, whereas solicited reports are those gathered as part of an ODCS. Both spontaneous reports and solicited reports in the ICH E2B electronic reporting standard can be designated as either being spontaneous or solicited. Solicited in that standard is called report from study in E2B. Section 4 of the guideline goes through the different sources of post-approval safety information that can end up as ICSRs. In particular, updates were made to the section on the literature, and these updates include harmonized guidance on the screening of medical and scientific journals, a clarification of when the reporting time clock, 15 days for expedited cases, starts for literature cases. The start of that clock is called day 0, and several other updates to help clarify expectations for marketing authorization holders in the management of safety data from literature. Again, I won't be going through every section in the guideline. I'm just picking out the key changes that were really responsive to the reasons that ICH formed the expert working group and set out to revise the guideline. And one of those reasons is digital platforms or the internet. This section replaces the internet section in the previous version of the guideline. Importantly, this section makes a distinction between how cases are managed based on whether the marketing authorization holder has responsibility for the content that's on the digital platform, which is the source of the information for the case, or whether they don't have responsibility for that content. So for example, a company's own product website would be a digital platform, of course, that they manage and they control the content. And so that digital platform would be considered under the MAH's responsibility. And the guideline provides a description of what that means, whether or not a digital platform would be under an MAH responsibility or not. As is the case in the original guideline, The revised version also goes on to say that marketing authorization holders have no obligation to screen digital platforms for which they don't have responsibility, or digital platforms that are external sites. However, if the digital platform is under the MAH's responsibility, they are required to regularly screen that platform in order to find and appropriately manage adverse events or adverse drug reaction information that may be posted on those platforms. Cases should be managed as either spontaneous or solicited, depending on the nature of the activity, depending on the context in which that data was collected, whether it was spontaneous or solicited. Well, MAHs aren't required to go out and actively screen digital platforms not under their responsibility, that is, external platforms. They do more and more frequently encounter post-approval safety data from these sites. The revised guideline provides some recommendations on how MAHs might go about managing the potential adverse events and adverse drug reaction information that they find on these kinds of platforms. MAHs can screen the data using an organized data collection system. Recall that an ODCS requires that the MAH have some basic documentation on their plan for their organized data collection. Using the ODCS, the MAHs should appropriately collect and manage adverse events and adverse drug reaction cases that they encounter while executing their plan for the digital platform data, but they don't have to actively go out and screen or search the entire data set to find adverse events or adverse drug reactions that may be somewhere in there in the data set beyond conducting their planned ODCS analysis. The revised guideline also clarifies the start of the time clock for regulatory reporting of cases identified from digital platform data not under the MAH's responsibility. And last, and perhaps most important, the guideline proposes the addition of a few new values in the E2B reporting standard. We're proposing that there'll be a new value, a flag, that will identify which cases were sourced from ODCSs conducted on a digital platform. All solicited cases are coded in E2D as report from study, but that includes any type of study, observational studies, registries, patient support programs even. And so this flag will allow regulators and industry to parse out which study cases were from this data source and digital platform, as opposed to a non-interventional study or a patient support program or some other ODCS. And the expert working group believes that the ability to identify this, which cases are digital platform, which cases are patient support program, will be of significant benefit for signal detection purposes. The next source discussed are patient support programs. The new definition for patient support programs provides some clarity and some guardrails on what programs can be considered patient support programs. This is a concept that's now widely used, but not really well standardized. The definition includes a few important concepts. First, all patient support programs are considered ODCSs. Second, to be a patient support program, a program must include collection of medical information or be designed such that the program will likely receive medical information. And last, this definition excludes programs that strictly involve delivery of a medical product to a patient's home, or which only give out coupons or discounts. These kind of programs don't involve the two-way type of interaction that's described by the collection of medical information, and so they don't qualify as PSPs under this definition. Because PSPs involve the interaction between the MAH and the consumer or healthcare provider, and adverse events and adverse drug reactions are typically collected by the MAH in the context of that interaction, the safety reports from PSPs are considered solicited or study reports. And again, there's a proposal in the new guideline to add a flag, a value in E2B that would identify cases for patients from patient support programs, and that would help in signal evaluation. And the last source of post-approval safety information that I'll mention are market research programs, or MRPs. As with PSPs, this definition is new. And again, MRPs are also all considered organized data collection systems, ODCSs, and cases from these kinds of data collection should be managed as solicited. And again, just like with PSPs and digital platforms, there is a proposal to add a new value in E2B that would identify which study cases came from MRP's market research program. Chapter 5 talks about what should be reported, and this section was updated so that it's more harmonized across regions. Some of these regions have revised their requirements for reporting adverse reaction data, particularly with respect to what things are considered, or what are the requirements for serious reports versus non-serious reports, or unexpected versus expected reports. And so the new description of what should be reported is adapted to be more harmonized across regions. The section also discusses what we call other observations. These are scenarios that in most regions are not considered adverse events or adverse drug reactions in and of themselves, and the guideline provides some clarified advice on what are the reporting observation, or what are the reporting obligations for these kinds of other observations, and in general, to report them only to a regulatory authority if it's required by the authorities in that local country or region. This is actually an exciting time for the E2D group. This draft was just approved by ICH and by the regulatory stakeholders to be published for public consultation, which is a significant milestone in the drafting of a guideline. All stakeholders can find the full guideline at the links presented on this slide, and can also provide their comments and feedback for the expert working group's consideration as we revise the guideline. Shortly, many of the individual regions will also publish the draft for comments in their respective regions and for comments from their respective public stakeholders. The public consultation period is expected to conclude this summer, at which time the expert working group will review and revise the guideline as needed based on the comments. The expert working group's work plan forecasts reaching a finalized guideline in May of 2025. So thanks again for the opportunity to present this update.
Good morning and good afternoon, everyone. My name is Carole Legaré, and I will be providing an overview of the ICH E6 R3 Step 2 document on good clinical practice. First, I would like to acknowledge that what I will present comes from the ICH presentation posted on the ICH website. The draft E6R3 was signed off by ICH on May 19, 2023, thus allowing us to proceed with public consultation. At this time, consultation has now closed. The initial version of E6 was published in 1996 and was updated in 2016. E6 should not be read in isolation. Many other ICH guidelines may apply, but E8 is especially important. This is because E8 discusses the clinical trial design issues that are most critical to clinical trial quality. After E6R2 was published, many academic researchers wrote to ICH to complain about a lack of flexibility and the lack of their involvement in developing a guideline that they are required to follow in many jurisdictions. The article shown here, for example, reports on the results of stakeholder interviews conducted by the Clinical Trial Transformation Initiative. 5 teams emerged from these interviews on participants' expectations for the revision. Incorporate flexibility, simplify the guidance, accommodate advances in research conduct, update and clarify language, and be transparent and inclusive. The E6R3 working group started by doing a gap analysis, looking at stakeholders' comments from various sources listed here, as well as other ICH guidelines. What we took away from the feedback was that although E6 is primarily intended for clinical trials to support market authorizations of medicinal products, it's also widely applied to other types of clinical trials of medicines. In fact, GCP is required for academic trials in Canada. There were also concerns that the R2 guidance had a one-size-fits-all approach with resulting over-interpretation of GCP requirements. There were concerns about the ability of clinical trials to meet all GCP requirements in different situations, such as those we saw during the pandemic. Clinical trials are a fundamental part of clinical research that support the development of new medicines or the new use of existing medicines. Well-designed and conducted clinical trials help answer key questions in healthcare and drug development. Their results are essential for evidence-based decisions. Trials with inadequate design or trials that are poorly conducted may place participant safety at risk and yield inadequate or unreliable evidence and are therefore unethical. They waste resources and the efforts and time of investigators and participants. The principles of GCP are designed to be flexible and applicable to a broad range of clinical trials. E6R3, along with E8, encourage thoughtful consideration and planning to address specific and potentially unique aspects for an individual clinical trial. Careful consideration of factors is needed for each clinical trial in order to ensure clinical trial quality. The development process for E6R3 was quite unique. In order to respond to stakeholders' requests for transparency and inclusiveness, we engage a group of academic stakeholders in a series of joint meetings with the expert working group. To increase transparency, we published the draft principles in 2021 and held public web conferences. There's also a plan for extensive training material to be developed using case examples to clarify the application of GCP. So what is new about E6R3 structure and content? We changed the structure of the guideline to provide more clarity and improve readability. We added language to facilitate innovations in design, technology, and operational approaches. We tried to set practical and feasible expectations around the responsibilities of the sponsor and investigator with respect to a digital environment. The guideline focuses on a fit-for-purpose approach with an emphasis on proportionality and risk-based approach. We incorporated lessons learned from the pandemic, and the guideline encourages public registration of trials and the disclosure of their results, and it also encourages the use of concise consent forms and the use of technology to support the consent process. The foundation of E6R3 is the set of principles. They would apply in all situations. Annex 1 describes the GCP expectations for clinical trials, and they would apply to all types of clinical trial. Annex 2, which is under development, will provide additional considerations for specific types of trials using decentralized trials, pragmatic trials, and the use of real-world data in clinical trials as examples. However, Annex 2 will not introduce any new requirements. Rather, it will discuss what needs to be considered so that GCP can be applied to those trials. Here is the revised structure. we have an introduction, followed by the principles of GCP. Then Annex 1 is made up of 4 sections which deal with ethics committees, investigator, sponsor, and a new section on data governance. Then we have a glossary and 3 appendices, one on the investigational brochure, one on protocols and protocol amendments, and one on essential records. The thought process behind the revision was to get people involved in the development of clinical trials to think about what is important, involve the right stakeholders, and design trials to answer the scientific questions while ensuring participant safety and the reliability of the results. It therefore builds on E8, which encourages a proportionate risk-based approach with a focus on activities that are critical to achieving the trial objectives. The guideline aims to support efficient approaches to trial design and conduct. It further expands on the use of technologies such as digital health technologies and wearables. It also encourages including the perspective of stakeholders such as patients and healthcare professionals in the trial design, which can lead to more meaningful trial outcome measures and guide decisions on feasibility of the study. Again, in line with E8, trials should identify factors that are critical to ensure participant safety and the integrity of results. The processes and risk mitigation strategies should be proportionate to the importance of the data being collected and the risk to the safety of participants. We should avoid unnecessary complexity. While there are changes in all sections of the document, the ones listed on top were the object of more substantial changes. Here's a table relating the revised principles to the sections of E6R3. The principles are not entirely new, but we put additional focus on responsibilities and risks. The principles on risk proportionality focuses on participant safety and reliability of results, and on risks that go beyond those associated with standard medical care. Given the blurring of lines of responsibility between investigators and the sponsor with the changing clinical trials landscape, the principle on roles and responsibilities focus on clarification of transfer of activities by the sponsor and delegation of activities by the investigator, and the maintenance of appropriate oversight. All the principles were revised to some extent. For example, one should make sure not to unnecessarily exclude particular participant populations. The informed consent process should take into consideration relevant aspects of the trial, the setting and context in which the trial will be conducted, and the potential use of technology. Also, periodic review of the scientific knowledge should be conducted to determine if modifications to the trial are required. The quality of the information generated in the trial should support good decision-making, and the protocol should be clear, concise, and operationally feasible. There is an emphasis that trial processes should support key trial objectives, and sponsors are encouraged to register trials in publicly available databases and disclose their results. Investigational products should be carefully managed and retain their quality throughout the trial. The first section in Annex 1 is on ethics committees. We included global language about reporting to ethics committees and regulatory authorities in order to reflect the different practices in different jurisdictions. We also updated the variable approaches to obtain consent and clarified the potential for participants to be compensated for costs incurred in order to participate in a trial. The next section is on the investigator. Again, it allows for various approaches for getting informed consent, highlighting that the ICF should be clear and concise and allowing the use of technology. It also clarifies that a process for reconsent should be considered if during the trial a minor reaches the age of legal consent in accordance with applicable regulatory requirement. The guideline clarified a number of points. For example, trial-related training for staff should be about their trial-related activities that go beyond their usual training and experience. The guideline also clarified the expectations between the sponsor and investigators regarding service providers. We clarified expectations regarding identification and maintenance of source records. For example, we should avoid unnecessary transcription steps in between the source record and the data acquisition tool. There should also be timely review of data that may impact participant eligibility, treatment, or safety. We clarified requirement for the documentation of delegation. For example, specific documentation of delegation may not be required in situations where the clinical trial activities are performed in accordance with routine clinical care. The next section is about the sponsor, which includes a new subsection on sponsor oversight. There are some clarifications under the monitoring section. For example, the monitoring strategy should consider the trial purpose, its design, the use of blinding, safety profile, and endpoints in line with the risk proportionate approach for that investigational product in that participant population. The monitoring plan should focus on aspects that are critical to quality. For the investigational product, it was clarified that alternative approaches for drug accountability records may be applicable when dealing with marketed products. We clarified that the sponsor should adopt a proportionate and risk-based approach to quality management, which involves incorporating quality into the design of the clinical trial, again a reference to E8, and identifying those factors that are likely to have a meaningful impact on participant safety and the reliability of the results. There are also clarifications that sponsor oversight extend to central facilities. There is a new section on data governance. It indicates that the sponsor should apply quality control to the relevant stages of data handling to ensure the data is of sufficient quality. The requirement for computerized systems should be proportionate to the importance of those systems and the data or activities they are expected to process. It addresses topics such as IT security, data protection, data validation, metadata, and data acquisition tools. The glossary section defines some new terms and revised the definition of others. Some definitions were added to support advances in the clinical trial ecosystem, while others were adapted to allow new clinical trial approaches such as decentralized trials. We also needed to ensure that they were media neutral in view of the greater role of technology. We also replaced the word subject by participant throughout the document and removed some confusing language. The first appendix is on the investigator's brochure. There was not much change to this section. We clarified that information on both the frequency and nature of expected adverse reaction should be included. The second appendix is on protocols and protocol amendments. We highlighted the importance of building adaptability into the protocol in order to avoid protocol deviations and amendments, as well as observations during inspection. In alignment with E8, we encourage the use of protocols that minimize unnecessary complexity and focus on what is critical. There is more information regarding withdrawal of consent. The appendix on essential records was the object of a major rewrite. E6R3 provides a list of criteria to determine whether a specific clinical trial record is essential and needs to be retained. We provided clarity on the management of these records. For example, for activities that are transferred or delegated to a clinical research organization, arrangements should be made for the access and management of the essential records throughout the trial and for their retention following completion of the trial. We created two tables, one outlining what will be considered an essential record in every clinical trial, and one listing potential essential records. So these are records that would become essential if they were generated during the trial. In summary, novel approaches to clinical trial design and conduct have the potential to streamline drug development and increase the convenience of clinical trials for participants. The intent of the revised E6 is to facilitate innovation in trial design and conduct while helping to ensure participant safety and reliable results. We are currently addressing comments from the consultation, and the final document is expected by the end of the year. Thank you.
Thank you for the great presentations. We have one more presentation before we take a 15-minute break. The final presentation before the break is on M14, General Principles on Plan, Design, and Analysis of Pharmacoepidemiological Studies that utilize real-world data for safety assessment of medicines by Dr. Melissa Kampman, Manager, Data and Analytics, Real-World Evidence Division, Marketed Health Products Directorate, Health Canada. Please join me to welcome Dr. Kampman.
Good afternoon, everyone, and thank you for the introduction. My name is Melissa Kampman, and I am a manager in Health Canada's Marketed Health Products Directorate. I'm very happy to be with you today to present an overview and update of the ICH M14 guideline that is currently under development on general principles on plan, design, and analysis of pharmacoepidemiological studies that utilize real-world data for safety assessment of medicines. As I'm sure most of you are well aware, epidemiology has long been a mainstay of post-marketing pharmacovigilance and safety evaluations. We as regulators have long recognized this utility and have written a number of guidelines and guidance to provide recommendations on the design, conduct, and submission of these studies for regulatory purposes. We also recognize that the availability of real-world data also continues to steadily increase worldwide, and that the role of real-world data in the therapeutic product lifecycle continues to evolve. That being said, there's currently no harmonized international approach here, and this may result in issues and inefficiencies for both sponsors and regulators. ICH has recognized that there are potential benefits to a harmonized guideline specific to the use of real-world data for the safety assessment of medicines, as it may result in decreased review effort, a decrease in the time until sufficient data is accumulated, or increase the likelihood of the transportability of studies and results across regulators. With that, I'd like to provide a brief overview of the efforts to harmonize the international guidelines into a single document, ICH M14. Please note that additional information on M14, including links to the concept paper, business plan, work plan, the group's rapporteur and regulatory chair, and a list of working group members are available on the ICH website. I'll first begin with some background and history. The proposal for a multidisciplinary guideline began with the development of an initial reflection paper on the need for harmonization. So this document focused on the technical requirements for studies, and it also outlined the benefits of developing a globally harmonized approach. It aimed to help facilitate use of real-world data in developing evidence for post-marketing safety regulatory work, and this document was endorsed by the ICH Assembly in June of 2019. To support this, a pharmacoepidemiology discussion group was formed. So this group, referred to as PEPIDG, was established in September 2019 following endorsement of the reflection paper to support the advancement of harmonization of scientific and technical requirements related to pharmacoepidemiological studies. The efforts of this group identified and harmonized requirements for these studies based on the most current scientific evidence, and a concept paper outline was developed that was endorsed by the ICH Assembly in July of 2021. Through their work, PEPIDG conducted a gap analysis to determine areas addressed across the greatest number of guidelines submitted by the 7 parties listed on this slide. So you can see here that this includes both US FDA and also Health Canada. 39 initial guidelines were reviewed and refined to focus on unique guidelines while excluding those that were deemed out of scope. 24 guidelines were then categorized into 8 topics based on their contents and included areas such as general principles, methodology, protocol and report formats, and regulatory purpose. It should be noted that some of the output of this group has been relied on by ICH M14 to identify the major topical areas suitable to harmonization. The discussion group's final concept paper laid out some of the goals of the harmonization effort from their perspective. So this included to increase confidence for pharmacovigilance activities using real-world data, to accelerate accumulation of safety data, and to promote sharing of post-marketing safety information among different regulatory agencies, so to lead to better decision-making. Finally, ICH M14 was formed in April 2022, with the expert working group being formalized in May of that same year. So M14 consists of 22 international groups, so you can see them listed here on the slide, that are working together on a harmonized document to describe the general principles on the plan, design, and analysis of pharmacoepidemiological studies for safety assessment of drugs, biologics, and vaccines. So next I'll provide a high-level overview of the draft guideline itself. So please note, as I mentioned towards the beginning of my presentation, that if you want to access additional information, that our concept paper and business plan are also available on the ICH website. In terms of draft guideline development, so far, the expert working group has taken all of the documents submitted from all members, and building on the work of PEPIDG, we have looked across them for common topical areas. We then harmonized the content of each of those areas to develop our first draft of the guideline. This initial draft was then circulated for review by all participating members in June 2023, where we received a total of 615 comments and proposed edits. So these comments and edits have been reviewed by the group, including in some cases a smaller working group, as well as including during a informal face-to-face working group meeting that was held in August of last year in Halifax on the sidelines of the International Society of Pharmacoepidemiology's annual meeting. So at present, the working group is now working towards ICH parties' consensus and looking towards the formal public comment period. Now, the focus of the draft guideline is on post-approval non-interventional studies on the safety of drugs, vaccines, and other biologics, so studies with treatment assignment are excluded. However, it should be noted that the principles may be applicable to these studies when real-world data elements are included. The principles may also be applicable to studies conducted for purposes other than safety evaluation. So, for example, studies that are focused on drug utilization or effectiveness. Now, on this slide, you will find a snapshot here of the table of contents for the draft guideline as it currently stands. So, in addition to an overview of general principles, it includes a framework for generating adequate evidence using real-world data. There is a section on initial design and feasibility that focuses on formulating the research question or questions and assessing feasibility. This is followed by a section on protocol development that includes principles related to study design, data sources including appropriateness, data types, standardization, and data quality and collection, the target or study population, exposures, outcomes, and covariates, addressing bias and confounding, and validation. The data management section includes principles related to the data management plan, quality assurance, and quality control. And the analysis section focuses on the analytical strategy, including considerations related to primary analyses, missing data, and sensitivity analyses. This is then followed by sections focusing on reporting and submission to regulatory authorities, dissemination and communication related to the study, including of the protocols and statistical analysis plans, and principles related to study documentation and record retention. And then finally, in addition to reference lists, including lists of both regulatory and non-regulatory guidance referenced, we have included considerations for specific or special populations, such as a focus on pregnancy studies. So, as I look towards concluding my presentation, I'd just like to speak to the current status of the guideline as well as next steps. So, as I just mentioned, the ICH M14 parties are currently working towards consensus on the draft guideline, which is targeted for April of this year. Once consensus is reached, a 90-day public consultation period will be launched through the ICH website. And so instructions on how to comment will be posted on the website at that time. And I would really like to take this opportunity to encourage you to take a look at the draft when it is released and to provide us with your comments. We very much value your feedback on this draft guidance. Finally, in terms of next steps, on this slide, you will see an overview of each of the next steps as well as targeted dates. So I would like to note that our guideline establishment goal date is currently set for early 2025 with implementation targeted for May of 2025. And again, additional information will be available from ICH as we progress through the different ICH process steps. So to conclude my presentation, I would first off like to thank you for your time. I would also like to thank our M14 rapporteur, David Money from US FDA, for his great work in leading the working group and his input to this presentation, as well as input from working group member Catherine Coet from the European Medicines Agency on the presentation itself. Now, before we head to break, I would like to once again encourage you to submit your comments during the public consultation period, and again, reiterate that we very much value your input. So please stay tuned for a future announcement once the consultation period is launched. I would also be happy to answer any questions you may have during the questions and answer period at the end of all of the presentations. So thank you very much again, and please enjoy the break.